Sequence homology. As we know that mutations accumulate in sequences and hence sequence diverge. But sections of sequences may still retain enough similarity so as to allow the identification of common ancestry or evolutionary origin. And that is the basis of protein structure prediction in this tutorial. Common evolutionary origin or sequence homology. But in this case, there are certain misleading or confusing terms. For example, sequence identity, sequence similarity, and sequence homology. These terms are quite interchangeably used, and so let us see what each of one of them actually means. First of all, let's talk a bit about sequence identity. So as the name suggests, identity means being identical or exactly same, and hence sequence identity means that the exactly same residues are being present at corresponding positions in the two protein sequences which are actually being compared. So we have the exactly same residue which is present in both the protein sequences. For example, presence of alanine residue at position 12th in protein sequence 1 and 2. That means it is contributing to sequence identity. Sequence similarity. Sequence similarity means we have similar kind of residues but not exact. That means it is quite different from identity, wherein we were having exactly the same residue present, but in sequence similarity we are having similar kind of residues. Now, what do we mean by similar kind of residues? For example, in protein sequences, what we can say is similar residues are those amino acids which have similar kind of physicochemical or maybe functional properties. So, if we take an example, if we have the substitution of aspartic acid and say glutamic acid by one another, so this kind of substitution in the two protein sequences will be regarded as a positive or a similar substitution. So it is not an identical substitution but a similar kind of substitution, because although the amino acid is different, one being aspartic acid and the other being glutamic acid, but they have similar physicochemical properties. For example, both of them are acidic hydrophilic amino acids. So I hope you got the meaning of sequence identity and sequence similarity and where these two terms must be actually used. The third term, which is actually of relevance in today's tutorial, is sequence homology. So, sequence homology in naive terms means having common evolutionary origin. Sequence homology is actually an evolutionary term that is being misused most of the times in literature, and it could denote either sequence identity or sequence similarity, but it should not be used quite interchangeably, because sequences must be called homologous if they have a common evolutionary origin, right? So it is as simple as that. If they are derived from a common ancestral sequence, they are homologous. So sequences are either homologous or they are not. Simply, they could not be any kind of percentage homology or percentage or we can say another kind of homology, a significant homology. So either we have to regard the both of the sequences as being homologous or not being homologous. That's the way. So, then how do we actually say that two sequences are homologous? Because there could be a point that we cannot actually go back to the point in time and then measure or then see the sequence testing that whether these two sequences are actually having common ancestor. and descendants, right? So we cannot go back in time. So how do we actually say that these two sequences are homologous? So we have two measurements or quantitative measurements by which we can say that there's homology between two sequences. First one is the sequences having high degree of identity. And the second measurement, the quantitative measurement, is that the sequences have high degree of similarity also, right? So the quantitative identity or else the quantitative similarity between two sequences is what we actually measure to conclude whether these two sequences are homologous or not, right? For example, if we take an example of a protein, the metalloprotein 1, which is a protein present in both rats and mouse. So this protein is actually 95% identical but 98% similar. So by quantitative measurement of this similarity and sequence identity, we can say that this particular protein, it is homologous for rat and mouse organism, right? So the correct term to use is homologous or non-homologous. We cannot use percentage homology or significant homology terms. They are quite misleading. So these proteins are said to be homologous.